Thank you, and it's very, very nice to be here with you. And after a very nice lunch, very good for your gut microbiome. So I'm going to attempt to give you a whistle-stop tour of the gut microbiome. And you're probably very aware there's a lot of media focus even on this at the moment. So it's a very early research area in the grand scheme of things. But there are indications that it is implicated in brain health and disease. And I'll give you a little flavour of some of the work that we're doing then in the context of Alzheimer's disease. So just to start then, microbes, microbiota and microbiome, these are just the terms that are used. So just to clarify, the microbes are the viruses, the bacteria and other microorganisms, or these tiny little entities. And bacteria are the most prevalent in the gut. The word microbiome then just incorporates the genetic material. And when you hear microbiota, that's really focusing on the microbes that are in the gut. So just in terms of clarification. So we have microbes living all around our body, not just in the gut, on the skin, the lungs, reproductive tracts as well, but predominantly in the gut. So just to give you an idea of the scale or of the microbes in the world, this is just some quick facts. There are more microbes on one person's hand than there are people on the planet. Yeah. So our bodies contain 100 trillion microbes. But interestingly, less than 5% of those actually cause disease. So our microbes are good for us. They keep us healthy predominantly. In terms of the bacteria on the gut, then, the bacteria in the gut can weigh over 4 pounds. So again, quite a lot. And evidence now shows that bacteria might influence our behavior and that there are also neurons in the gut. So a lot of evidence to suggest that there is this connection between the gut and the brain. And if there are that many microbes around, they must be doing something. So what are the functions then of these gut microbiota? Why do we have them? Well, they're involved in digestion of our food. And so particularly fiber. So if we digest fiber, it releases these molecules, including what are called short chain fatty acids. And these are very, very important for controlling inflammation in the gut and in the body. So then associated with that then is the protection that our gut has against pathogens, and pathogens being the bad microbes. The gut microbiota also produce vitamins like vitamin K and vitamin B, some of the vitamin Bs, which control our energy levels and also control our cholesterol and blood sugar levels. So very important functions for the gut microbiota. So where do they come from then? Well, when we're born, before we're born, we're in a sterile environment. And then when a baby is born, they will acquire the gut microbiota through the birth canal from their mother, and then through breastfeeding, bottle feeding, etc. So then up to about age 3, when a child is on an adult diet, essentially, there is a pretty stable gut microbiota present. But it's modifiable by diet in particular. As we get a little bit older then, the diversity and the abundance of the microbiota can decrease a little. So what we mean by diversity is a reduction in the different types of the microbiota. And this can be due to malnutrition, for example, or malabsorption, which can happen and occur as we get older. But overall, people that have a greater diversity in gut microbiota, that means more different types, tend to have better health. So this is just to show work that was done by colleagues in Cork over 10 years ago now, to show that the gut microbiota composition correlates with diet and health in the elderly or in the older population. So really, those that had a decreased diversity of their gut microbiota tend to be more frail and indicators in that regard. So since then, there's been a lot of work done to show that the gut microbiota might be involved in systems like the immune system, cardiovascular system, involving the heart, the gastrointestinal tract, or the gut, and the brain. So you might have heard the term the gut-brain axis or the microbiota gut-brain axis. So what this is really is a communication pathway, or it's called a bidirectional two-way pathway between the brain and the gut. But in actual fact, there are 4 pathways. So more and more evidence coming out to show these connections. So the nervous system, particularly the vagus nerve, so this is a large nerve that controls our digestion, our breathing, heart rate, and other functions in the body. The immune system releases these tiny entities called cytokines, which are soluble in the bloodstream and the gut and can access the brain. The endocrine or the hormone system, also hormones can communicate between the brain and the gut. And microbiota products. So the little microbes themselves can release metabolites into the gut, into the bloodstream. And more evidence now is coming out to show that these metabolites can influence brain function. So really the thinking is, if you have a healthy gut, you have healthy brain function, and in terms of behavior, cognition, emotion, etc. And indicators and studies have shown that if there is an altered gut microbiota, such as in diseases or in response to stress or in a negative environment, that it can influence our brain function. So again, we're just proceeding with caution because this is a very early area of research, but indicators are there. Okay, so if we have a think then and have a look about what are the indicators or what have been shown in terms of gut microbiota and Alzheimer's. So it's really in Alzheimer's where most of the work has been done as opposed to other types of dementia. So a number of groups now have done work on this, and there's been various meta-analysis. And the consensus is that there is quite a decrease in the diversity of the gut microbiota in Alzheimer's patients. So if we drill in a little bit into it and say, okay, well, what microbes or what bacteria in particular have been shown? So the main players are these firmicutes, it's called. So these are a beneficial bacteria. And these have been shown to be, the abundance of these have been shown to be decreased, while bacteroidetes are an inflammatory type of bacteria, and the prevalence of these bacteroidetes have been shown to be increased. So interestingly, the bacteroidetes, these bacteria are more prevalent in the Western world and are linked to diet, like what we call a Western diet, like high fats, high sugars, etc. So it's interesting then that that's kind of more of an inflammatory type of diet that can occur. So these associations have been shown between the types of gut microbiota. So this is if you're a microbiologist, these are the level of the phyla, but they're also the level of the genera, and there's a lot of complexity here around that, showing the changes. But these are the associations that have been shown in the patient population. But we don't really know if the changes that occur in the gut can actually cause changes in the brain or in the behavior or in the symptoms. And really, that's a really difficult one to solve or to figure out. So the question is really, do these changes in gut microbiota, can they cause any changes in the brain or in behavior or in the symptoms associated with Alzheimer's or with dementia? So a number of years ago, different approaches to investigate how we tackle these type of questions have come about. And fecal microbiota transplantation, or FMT, you might have heard of this, is one approach. So just taking you back, this is a medical procedure, and it's used and it's only approved for the treatment of C. difficile or colitis, right? So what this is where a microbiota sample in the form of a stool sample is taken from a healthy individual, it's filtered, it's processed, and then delivered to a patient, let's say with colitis, and this is via colonoscopy or orally through capsules. So this technique, if you like, or can also be used in an experimental setting. So if we're asking the question, can you transfer, if you transfer microbes from an Alzheimer's situation to a healthy situation, can you transfer some of the changes, the symptoms? Okay. Now it wouldn't be ethically to do this from human to human. Okay. So it wouldn't be ethical to take microbes from an Alzheimer's patient and give them to a healthy person. So what we do is we do it in animal studies. Okay, so taking a fecal sample from human Alzheimer's and put them into mice or rats, and then we can assess the behaviors, the cognitive, the memory behaviors in these animals to see then, is it possibly through the gut microbiota that changes might be coming about. The other thing in the context of when we look at brain changes, we kind of look at the resolution of the neurons in the single cell in a human brain. The MRI technology, for example, just really isn't there to look at the level of the cell, but we can do this in animal research. So that's where it plays a role in this regard. Okay, so back to our question. Does fecal microbial transplant from Alzheimer's patients to healthy young rats cause changes in behaviors and neuron function associated with memory or with dementia or Alzheimer's? So this is the question we asked, and it felt a bit nuts at the time doing some of this work. But we had collaborators in Italy, and they had recruited cohorts of Alzheimer's disease subjects and age-matched controls. And so we took those samples, those stool samples from those patients, and Stephanie Grabroker, she's a postdoc in the group, she transplanted these into rats. And she did a series of behaviors, not just the cognitive memory behaviors, but also some depressive anxiety behaviors. And these all have been designed in animal studies to mirror as much as we can what happens in the human situation. There's always going to be limitations with these. Okay, so what Stephanie found then, and what we found when we did the work was, first of all, when we looked at the human stool samples, the microbiota, we saw that these verticutase, these beneficial bacteria were reduced, and the inflammatory bacteria were increased. And we also saw this in the microbiota of the rats after they got the transplant. Okay, so that was transferred over. When Stephanie did the behaviors, it was really only the memory behaviors that there were deficits and that there were changes in. So the episodic type memories that we can measure in the animal situation, spatial memory or pattern separation, these memories were impaired in these rats. We also looked at some of these metabolites. So these are what are released from the microbiota into the gut area and in the brain. And we saw that there was a decrease in some of these, and especially those that are involved in memory function and brain health. So indicators here that there must be something in these gut microbiota after all. Having a look then to see what happens in the brain. So it's been mentioned earlier about the hippocampus having a role in memory and being affected and impaired early on in the disease in dementia. So in the hippocampus, there's a category of neurons, of new neurons, and that they've been shown to regulate some of the types of memory that are implicated in Alzheimer's. So these new neurons are constantly being born in the brain, and the word neurogenesis means neurons being born. So we looked at this process in the brain. And a few years ago, there was a really very interesting paper that was published, and it showed that these new neurons in the brain, they decrease with age, but there's a huge decrease then in patients with Alzheimer's disease. Okay, so we think maybe these types of neurons are implicated. So what we did is we looked at the brains, and we do this using microscopy techniques in the rats, again, after they have this transplant, and we can look at the neurons, we can look at their growth, we can look at their development to see how healthy they are. And we did see that there was a decrease in an impairment, if you like, in this neurogenesis, in this neuron growth. So putting this information together, Alzheimer's symptoms can be transferred by the gut microbiota, at least into the rat, suggesting, and I say suggesting, because we're really only tipping the surface on this, that the gut microbiota may have a role to play in the disease. There's a lot more going on in dementia and Alzheimer's, as you know, but this is just one set of experiments that implicates perhaps maybe the gut microbiota might be doing something. So how can we use this information then? Well, you've seen this slide before around the modifiable risk factors associated with dementia. So I just highlighted one here. This is one of the new ones that's been added just this year, the high cholesterol levels. So if you remember when we spoke about the functions of the gut microbiota, well, the gut microbiota can control our cholesterol levels, and they also can control our blood sugars. So this is linked to diabetes, which is another one of these modifiable risk factors that are have been shown in associated with dementia risk. But it's not just these two, because the whole world of the gut-brain axis and gut microbiota has been exploring different types of influences on the gut microbiota. So, for example, diet, mode of delivery, the environment, drugs, exercise, all of these impact the gut microbiota, and there are effects of these changes as well. So I've just highlighted here some of these risk factors linked to dementia risk, which are also implicate the gut microbiota. So it's just an observation, really, that maybe there is a link here. And the reason is that the gut microbiota are very responsive to our lifestyle, our environmental factors. Okay, so they could be potentially a route through which the changes that we experience in our lives and our lifestyle factors could affect the gut microbiota, which consequently affect how our brains are and how we behave and our symptomology. Okay, so just as one of the strongest indicators of the effects on the microbiota is diet. So again, you'll have heard this again through the media, but the main messaging is that foods that are rich in fiber, like whole grains, veg, fruit, or the omega-3 fatty acids, phytochemicals, and probiotic foods. These are fermented foods like yogurts, etc. They have microbiota living in them, so to speak, and they help the good gut microbiota to expand and increase the diversity. So these foods have been implicated to improve or develop good gut microbiota. And these food constituents are part of what we call the Mediterranean diet. You may have heard of this, and there has been work in the UK and in Queen's and Belfast as well to show that Mediterranean diet adherence is associated with lower dementia risk. And actually, the World Health Organization recommends then a Mediterranean-like diet to help reduce the risk of cognitive decline and dementia. Okay, so associated with these modifiable risk factors. Exercise is another one, and Catherine will speak to that as well. So it's been shown that exercise can increase the diversity, again, types of gut microbiota, and that those that are leaner in older age and that are healthier and physically fitter tend to have a better microbiota. So we have some work going on in this as well, looking at the differences, for example, in males and females and how they exercise and respond. So just to kind of finish off really is to show you that looking at this schematic again about the modifiable risk factors, and where most of them are indicated is in the midlife period of the lifespan. So in middle age, which many of us are interested in at the moment. And so a lot of changes occur in the gut microbiota in middle age, but also in the brain. And we're very responsive to how the environment influences. So we might brain age healthily or not. So middle age, then maybe this is really our window of opportunity to enhance our gut and brain function. So again, just a pointers towards some of the work we have ongoing with collaborators in London, where we're looking at the role of the gut microbiota in changes in memory, mood, and these are in human cohorts, that middle-aged humans that are exercising, and we're looking then at the gut microbiota and the implications in memory and mood. So really just want to leave you with this slide, because while we can do these experiments and while we can point to maybe individual factors of the gut, etc., there isn't one size that fits all. And so I think we need to think about this when we are, attending to our health and reducing those modifiable risk factors. We need to think about the complexity of the human situation and how we all live in different ways. And we need to find what works for us in terms of maximizing our health, maximizing our gut and our brain health. Thank you.